Hello, it's a pleasure to be speaking in this PMD Family Symposium, and I want to thank the organizers. My name is David Rowich. I'm a professor of pediatrics at the University of Cambridge and also at UC San Francisco. Today I'll be speaking about cell-based therapies, patient models, and new treatments for Pelizaeus-Merzbacher disease, or PMD. The disclosures I'd like to mention is that I was an unpaid consultant for Stem Cells Inc., who is the sponsor of the phase 1 clinical trial, and other companies I consult with are listed. Families often describe their children with PMD as being trapped in a brain without myelin. Myelin is an essential substance for communication of the nervous impulse, and of course in PMD, because of mutations of PLP1, myelin is never produced in an effective manner. Boys fail to sit, crawl, walk, or speak, and often death can be premature in adolescence. We can use MRI to measure myelin in the white matter, and shown in this image is diffusion tensor imaging that shows the ways that myelin connects the brain, performing an essential mode of communication between brain centers. How can we help PMD patients? Well, we've heard about some approaches earlier in the symposium from other investigators, and generally we think about how we can replace or repair defective endogenous oligodendrocytes, the myelin-producing cells that carry a PLP mutation. Also, we can measure their outcomes clinically and also by using MRI, which I showed you as a way that we can measure how myelin can promote transmission of brain communication. There are three potential ways forward. First, we can think about fixing the mutant PLP gene or gene therapies. Second, can we find drugs that can help the PMD oligodendrocyte work better to produce myelin? And third, can we replace the defective oligodendrocytes by transplantation? I was principal investigator of a first-in-human study that used HUCNS, a neural stem cell product made by Stem Cells Inc., directly transplanted into the brains of boys with PMD. Shown in this image is the proof of concept that the cells, once transplanted into the hypomyelinating shiver mice, can engraft in white matter and produce myelin. And myelin is shown here in the fluorescent green image called MBP, and also by electron microscopy. So this was essential data that went to the FDA for approval of this first-in-man study. This shows the procedure and MRI findings at 1 year post-transplant. We injected 75 million HUCNS cells into 4 sites in subcortical white matter of 4 PMD subjects and followed them clinically and by MRI each year for 5 years in total. The cartoon shows the sites in which the cells were injected into the brain, and then lower left shows how we compared the impact of the transplant using MRI. We compared the sites that were injected, shown by the red arrow, to a control region of interest shown by the sort of turquoise box. This was not injected with cells, and we would expect those areas to follow the normal trajectory of development in a PMD subject compared to the areas that were transplanted. And what did we see? Well, over the first year, we are showing in the lower right-hand corner the measurement of diffusion tensor imaging by MRI, which shows an increase in the fractional anisotropy and decrease of the radial diffusivity of the areas that were transplanted with the neural stem cells compared to the control regions. And these components of FA and RD are those that you would expect to see associated with myelination of the axons. Here are images at 4 years post-transplant, and the white arrows are pointing to what we call hyperintense areas. They look white on a T1 image, and these would be areas that had been injected with the neural stem cells, showing that they are still demonstrating a signal after 4 years post-transplant. Moreover, these areas also showed increased fractional anisotropy and decreased radial diffusivity, properties we would associate with myelin. Next slide summarizes the clinical findings over the course of 5 years. We found that delivery, immunosuppression, and the human CNS stem cells themselves were safe over the course of the 5 years. 2 of the 4 patients developed class II alloantibodies against the graft. We did not detect tumor formation or inflammation, and the persistent MRI findings suggest that the cells had engrafted, but do not provide definitive evidence of myelin formation. And other limitations of the study is that it is a phase 1 study. It could not determine efficacy, and there was no control group. So this would normally have led to a phase 2 study, but unfortunately, Stem Cells Inc. went bankrupt, preventing us from doing the clinically efficacious test of neural stem cells. So where else can we take this problem? How else can we further innovate this? Well, we're in collaboration with Marius Vernig, and actually at the suggestion of a PMD family member, took it into the laboratory. And we derived from our patients in this study what we call patient-specific induced pluripotent stem cells. The next slide shows a cartoon for how induced pluripotent stem cells can be patient models of disease. We start with fibroblasts. We go through a process called reprogramming, which will induce pluripotent cells that can then be differentiated into specific neural cell types, such as oligodendrocytes. Moreover, we can gene correct those cells so that we can correct the PMD mutation, providing a control against which we can measure the impact of a PLP mutation. And then using these cells, we can put them through drug screening for drugs that might make the cells work better. And the animation shows that drug screening process. Next slide shows the summary results, which have been published last year in Cell Stem Cell. We used transcriptomic approaches to look at the genes that were dysregulated in PLP mutant cells compared to the gene-corrected controls. We found that many of the genes that were abnormal in the PMD oligodendrocytes were in the iron metabolic pathway. Genes that encode for proteins that handle iron or may be used in gene regulation that are iron regulators, such as IRP1. These were clues iron could be a culprit in PMD, and suggested the possibility that we could use iron chelators, which reduce the environmental exposure of iron, as a way of rescuing the PMD phenotype. The animation shows the impact of iron chelators against PLP mutant oligodendrocytes, or those that have had gene correction. And what you can see in panel A is a very unhappy looking oligodendrocyte. It is showing a very small size and very non-complex morphology. Compare that to panel B, where you see large oligodendrocytes that are spreading their processes out into the culture medium. The cells that were PLP mutant and did not have benefit of chelation actually went on to die through apoptotic cell death mechanism. But interestingly, we found that when we cultured them in the presence of iron chelators, like apotransferrin, shown in panel E, we restored the survival and also the complex sort of bushy morphology of a oligodendrocyte that was indistinguishable from that had gene correction. And this led to the idea that there could be also a clinically relevant translation of this finding, because deferoprone, a blood-brain barrier penetrant small molecule iron chelator, is available both in the US with FDA approval and also in the NHS. Next slide shows new directions, another new direction that we are taking to help identify PMD patients earlier. Often there is a diagnostic odyssey in which months or years may go by before a diagnosis of PMD is made. In the UK, we are using whole genome sequencing when a child presents to their doctor to come to that conclusion much quicker. And the slide shows 2 studies published in Cambridge last year and this year on large populations, showing the feasibility of whole genome sequencing as part of routine NHS practice. The next slide summarizes the findings from this talk. Firstly, I've reviewed the safety and MRI findings of neural stem cell transplant for severe PMD at 1 and 5 years. Secondly, I've described how in NHS we are using whole genome sequencing for earlier detection of PMD patients, and we think that could make it possible to enroll them sooner in relevant clinical trials. And thirdly, I've covered data showing that oligodendrocytes with PLP1 point mutations or duplication are rescued by gene correction or iron chelation. And this topic will be taken up subsequently in Dr. Nicole Wolf's talk. The final slide is acknowledgements, and I want to make mention of Nalan Gupta and the clinical team at UCSF that performed the clinical trial, Hiroko Nobuta in my lab, who worked with Myris Vernig at Stanford on the induced pluripotent stem cell work. And finally, I'd like to thank the PMD families for participating in research and your encouragement in our studies to identify new treatments. Thank you very much.